Genetically modified organisms (GMOs) can be defined as organisms in which the genetic material (DNA) has been altered in a way that it does not occur naturally by mating or natural recombination. Genetic engineering processes are usually based on the knowledge derived from microbiological research or genetics, and allow for specific intervention into the genotype of human living organisms and viral genomes. The genetic modification of organisms or genetic engineering can be divided into following four main categories. Green genetic engineering or agrogenetic engineering aims to develop genetically modified plants in agriculture or the food sector. Green genetic engineering is almost exclusively used in plant breeding. At present, most genetically modified plants cultivated worldwide have been modified to make them resistant to insects or tolerant to certain herbicides. Other breeding goals including increasing the yield potential, improving resistance to disease, heat, drought, cold or floods, improving product qualities, for instance concerning vitamins or fatty acids, and developing plants that are more suitable for production of industrial raw materials or that can be used in production of medicine. The second category is red or yellow genetic engineering, utilized in medicine, genetic tests, and gene therapy, as well as development and production of drugs such as insulin and vaccine. It is the most powerful and promising application of genetic engineering involves the treatment of genetic disorders like sickle cell anemia, Duchenne muscular dystrophy, cystic fibrosis, Tay Sachs disease, and Huntington's chorea. Now, medical scientists can identify more than 3,000 disorders happens because of the error in individual DNA. By this technique, scientists can modify the genome of organisms. The third category is gray or white genetic engineering. This is the production of enzymes or fine chemicals for industrial use with the aid of genetically modified microorganisms, such as the development of products for enhanced watching performance. The last category is genetically modified animals utilized for specific food production, such as dairy cows are modified to produce allergic-free milk.
Then, in this section, we will talk about the methods used in genetic modification. There are three most common methods of genetics modification that is most used, which is plasmid method, vector method, and biolistic method.
It has been argued that public health could benefit from using GMOs, and there are some issues which could be solved with current biotechnological instruments. So the first benefit is producing edible vaccine or medicine in milk, eggs, or fruit. The ability to genetically modify animals in order to produce pharmaceuticals in their milk has been one of the most innovative applications of genetic modification techniques. Medicine or vaccine produced in milk could be manufactured and distributed cheaply and made more accessible to people around the world. Injected vaccines are expensive, require trained medical staff for their administration, and usually require constant cooling during transport and storage, which create difficulties in many developing countries. The use of needles also brings the risk of spreading infection. Edible vaccine would help to avoid these inconveniences and dangers. There are examples of transgenic plants that have been developed to immunize against the hepatitis B and norovirus, both of which are substantial concern for individuals living in developing countries. Researchers have also produced a variety of transgenic potatoes that contain a small portion of cholera toxin and immunize against the disease upon ingestion. The second benefit is improving the qualities of certain crops and producing safer food. It has been reported that the use of some unconventional varieties of crops can be grave health consequences. Research on genetic modified crops or GM crops could create safer variety of these and other crops that would replace harmful traditional varieties by reducing the level of undesirable substance including mycotoxin, alkaloids, and glucosinolates. Breeding wheat increased yield while reducing the use of pesticides, improving plant adaptation to unfavorable environment. In order to achieve this advantage, herbicide-tolerant and pest-resistant GM varieties as well as virus and fungus-resistant crops have been developed. GM technology has also been used to generate crops that are tailored to particular environment. Such as drought-resistant varieties of crops that are tolerant of high soil salinity. GM crops may offer solutions to very specific climatic conditions prevalent in developing countries and allow for more effective control of pests and fungal infection. Some GM improvement may also additional benefit. For example, GM rice in China require less pesticide, sparing in addition to increased crop yields. Genetically modified bacteria may also able to serve as a barrier by secreting protein, protecting women against HIV infection. For example, a natural component of vaginal microbial flora Lactobacillus jersoni has been genetically modified to secrete soluble CD4, a protein that HIV specifically bind in order to gain access to cells and infect them, and has been shown to block laboratory strands of HIV from infecting humans. Malnutrition is a common third world country where impoverished people rely on a single crop such as rice for the main staple of their diet. However, rice does not contain adequate amount of all necessary nutrients to prevent malnutrition. Future GM crops could also have substantial direct nutritional or medical benefit to consumers. Crops could, for example, be genetically modified to produce micronutrients vital to human diet. One type of such of crops is golden rice, genetically modified to produce beta-carotene, the precursor to vitamin A. According to WHO in 2005, 45 and 122 countries have vitamin A deficiency of public health significance based on prevalence of night blindness and biochemical vitamin A deficiency in preschool age children. Genetically modified golden rice is one of the promising strategies to solve the vitamin A deficiency.
Next, we go into the disadvantages of genetic modification. There are many disadvantages of using genetic modification product, such as it is a technology that can be easily abused. We are currently have laws and treaties in place to prevent genetic engineering abuse. That doesn't mean it won't ever happen. The reality of genetic engineering is that DNA insertion could be used to create severe problems for certain groups of people. Imagine that someone is allergic to shellfish. Someone could insert shellfish in DNA into regular crops like corn. The person with allergy would eat the corn and potentially have an allergic reaction trigger because of it. Over time, we could also take approach we have to altering plants and animals to altering humans. If done, the consequences to our society would be numerous and unpredictable. Next, it creates difficult legal liabilities with unintended consequences. It isn't just DNA sequences that can be patented through genetic engineering practices. Seeds and crops can also be patented. That has caused issue for farmers who live near fields where genetically modified crops have been grown. The crops which have been genetically engineered have their seed spread to other fields, causing unintended growth where they land. Numerous property owners have been ordered to pay royalties and compensation for product loss because of this issue in Europe and North America, because of the patenting process. Because of this liability treat, few farmers want to work the field because it could cost them more than what they might make. Last but not least, it can have unintended negative consequences. Genetic engineering may be a proven science, but outcomes are not always predictable. Dolly the sheep is created as being the first mammal clone from an adult somatic cell. What is not often publicized is that Dolly was the only lamb that was born out of 277 attempts at the cloning process. There were only 29 early embryos created and 13 surrogate mothers were used in the effort to create Dolly. Genetic engineering can be very destructive when it wants to be, and the attitude towards the outcomes that are possible is that the ends justify the means to get there. That can be problematic when considering genetic engineering for human-based purposes. Hello everyone! In this section, we are going to talk about the recent technology of genetic modification. In this field of genetic modification, many technologies have been introduced to the world. For example, genetic modification organism, GMO, which is very popular in agriculture. It helps the farmer increasing the crop yield, also help them produce enough food for the people in the world as the population is increasing year by year. In 2050, the population is estimated to be increased by 33%, which is 10 billion. So, let's move on into the recent technology of genetic modification. The most recent genetic modification technology that have been introduced in the world are genome editing and CRISPR-Cas9. Genome editing, also called gene editing, is a group of technology that gives scientists the ability to change an organism's DNA. These technologies allow genetic material to be added, removed, or altered at specific locations in the genome. Several approaches to genome editing have been developed. A recent one is known as CRISPR-Cas9, which is short for clustered regularly interspaced short palindromic repeat and CRISPR-associated protein 9. The CRISPR-Cas9 system has generated a lot of excitement in the scientist community because it is faster, cheaper, more accurate, and more efficient than other existing genome editing methods. CRISPR-Cas9 was adapted from a naturally occurring genome editing system in bacteria. The bacteria capture snippets of DNA from invading viruses and use them to create DNA segments known as CRISPR arrays. The CRISPR arrays allow the bacteria to remember the viruses or closely related ones. If the viruses attack again, the bacteria produce RNA segments from the CRISPR arrays to target the virus's DNA. The bacteria then use the Cas9 or a similar enzyme to cut the DNA apart, which disables the virus. The CRISPR-Cas9 system works similarly in the lab. Researchers create a small piece of RNA with a shortcut sequence that attach to a specific target sequence of DNA in a genome. The RNA also binds to the Cas9 enzyme. As in bacteria, the modified RNA is used to recognize the DNA sequence, and the Cas9 enzyme cuts the DNA at the targeted location. Although Cas9 is the enzyme that is most used, the other enzyme, for example CTF1, also can be used. Once the DNA is cut, researchers use the cell-on DNA repair machinery to add or delete piece of genetic material or make changes to DNA by replacing an existing segment with a customized DNA sequence. Genome editing have a great interest in prevention and treatment of human diseases. Currently, most research on genome editing is done to understand diseases using cells and animal models. Scientists are still working to determine whether this approach is safe and effective for use in people. It is being explored in research on a wide variety of diseases, including single gene disorders such as cystic fibrosis, hemophilia, and sickle cell diseases. It also holds promise for treatment and prevention for more complex diseases such as cancer, heart disease, mental illness, and human immunodeficiency virus (HIV) infection. Ethical concerns arise when genome editing using technologies such as CRISPR-Cas9 is used to alter human genomes. Most of the changes introduced with genome editing are limited to somatic cells, which are cells other than egg and sperm cells. These changes affect only certain tissues and are not passes from one generation to the next. However, changes made to gene in egg or sperm cells or in the genes of an embryo could be passes to future generations. Germline cells and embryo genome editing bring up several ethical challenges, including whether it would be permissible to use this technology to enhance normal human traits. Based on concerns about this ethics and safety, germline cells and embryo genome editing are currently illegal in many countries.
Genetic engineering of crops is a new technology in its embryonic stage. It is essential to list any and all concern about commercializing genetically modified food. Genetically modified food has the potential to solve many of the world's hunger and malnutrition problems and to help protect and preserve the environment by increasing yield and reducing reliance upon chemical pesticide and herbicide. While the environmental, health and economic risks of GM crops should be carefully studied before full-scale adoption. The types of GM crops that are already available have thus far largely proven to be beneficial to agriculture and even to environment. Despite the opposition, the future looks bright for many GM crops and being able to successfully track and assess them will likely mitigate the GM food debate. Overall, there is increasing evidence to back up the argument that GM technology, new possibility for many types of GM food could be in the future. Agriculture innovation might finally move beyond providing commodity crops and fulfill the early promises that GM food will help feed the hunger and malnourished of our growing world.